Mr. Lawton, please come up to the stage. Give him a round of applause. Great, your 7 minutes starts now.
Good afternoon. I'm Eli Lyons, co-founder and CEO of Genomeminer. And I think my presentation might be a little different from the others in that I'm going to describe some of our business development challenges in the beginning and explain how they are related to our technical development. In my experience, for a small company, sometimes the business development can be just as important to overcome as technical challenges. So when we started the company, we just wanted to develop software, but that was very difficult to do on its own without a lot of showcase data. And in the end, we ended up developing some microbe products, which I'm going to explain on the next slide. And now we're just starting a human therapeutics program. And what these have in common is that we start with genome data first. So we mine genome data to develop these products. That's why we're called Genomeminer. And that includes using machine learning for this microbe product development. So as I mentioned, in the beginning we wanted to develop software, but we didn't have our own data, so we had to get our hands dirty. And we did that by going to Okinawa, and we started collecting microbes from the soil and screening them and then sequencing them at Okinawa Institute of Science and Technology. Now we have 2 microbes under feasibility study with a major chemical company in Japan to develop a biopesticide product. But of course, agricultural chemicals are a little bit low risk and low margin, especially for this audience. And so we want to do something with better margins, but also more challenging. So now we're also doing a screen of pet antibiotics using targets that are, again, provided by a much larger company. And in this case, it's challenging because we need to not just identify the microbe, but the chemical that the microbe produces. And finally, we're just starting a human therapeutics program, which I will also get to later. So in terms of our company partnership structure, we're developing the seed level products. And so we're looking for partners that want to bring these products to final commercialization. For example, for microbe, there's a lot of scalability issues, distribution issues, et cetera. We are not interested in doing that. We want to develop the seed level products for companies to bring to market. And for human therapeutics, we don't have a track record of partnership yet in that area, but we're looking at co-development of early stage products for oncology, specifically for fusion RNA targets, which I'm going to get to later. And this all, again, fits with bioinformatics. So I'm going to explain that a little bit. At our company, we eat our own dog food, meaning we collected the microbes and we sequenced them and we analyzed them on our own platform. And so that allowed us to have nice showcase data early on. Microbe genomes, of course, are not as complex and the data is much smaller than human genomes, but we needed something to get some traction. And we use this software really efficiently, and we can avoid buying servers, we can avoid hiring IT staff, and we automate and centralize a lot of data management. And now we have one of the largest, actually the largest pharma in Indonesia using our software. They're using it for SNP analysis, SNP invariant analysis. SNP invariant analysis can be used for drug identification. It can be used in the clinic, patient stratification. This particular company, their R&D team is using it for stem cell research. So in this case, they're doing some genetic engineering of stem cells, and they want to see what kind of variants it causes when they engineer these stem cells. So when their team uses the software, they just see this user interface and this user interface. So when they want to run a SNP analysis, they have like a little tab that has their proprietary pipelines. But this proprietary pipeline is actually running quite a complex analysis in the cloud in a fully automated fashion. And then we have developed and customized this pipeline for them. So this runs in Google Cloud. And how it works is that once they start the pipeline, on the back end, each step of the pipeline is spinning up a server separately with its own runtime environment and its own hardware requirements required specifically to run each tool in the pipeline. I want to note just a typo here. This should be 24 core, 128 gigabytes. So this company, by using our software, they don't need to buy any GPUs. They don't need to set up any servers. Everything is super stable. All the metadata from the analysis is saved. It's unmutable. So when they go back, they can see which version of the tool was run. They can make sure all the data is managed correctly. They can see who ran the pipeline, on what data set. Everything is managed. 1 minute, okay. And then at the end, they can use our genome viewer to actually see these variants in the genomic context of interest. So which genes is this actually affecting, and then plan their next experiment. Okay, so what we're working on now is how can we drug fusion mRNA targets. So these are typically undruggable or extremely difficult to drug targets. They're caused by rearranged genomes that cause gene fusions, fusion mRNA, which leads to fusion proteins, which are often a driver of cancer. And so we're targeting fusion mRNAs. And this would require, of course, a companion diagnostic that would be bioinformatics based. And so that's the key there. Our initial strategy is against lung cancer. Clinical use would be for low non-responders to immunotherapy, but we're also considering a direct cancer treatment strategy. This is super early stage. We do have some data that I can talk about in the question and answer section. My background, I came to Japan as a PhD student. That's where I met Paul and my other co-founders who were studying or working in Tokyo. And so that's how we founded the company here. And thank you, and I look forward to some questions.
Thank you so much, Mr. Lyon.